Okay, thank you very much. Nice talk. Um, we go on to the last talk, but not least, by Nuria. Nuria Izquierdo. So Nuria comes from, she works in IRSI Caixa, is an associate researcher in IRSI Caixa. And he's going to talk us about cyclic 1, how cyclic fuels HIV transmission in lymphoid tissue. Thank you, Nuria.
Hello, everyone. Thank you very much for staying to the basic session. It's great to have you here. And I would like to start by thanking the organizers for their kind invitation and also for giving me the opportunity to present some of the work that we have done at the AIDS Research Institute, IRSI Caixa. So, mature dendritic cells are the most potent antigen-presenting cells that we can find in our organism. And these cells mainly exert their functions within the secondary lymphoid tissues. As we all know, this is a key place for HIV replication. It's been long known, however, that mature dendritic cells can capture and store large amounts of HIV, and that these viruses are able to escape the classical degradation pathways that would normally lead to antigen presentation. And in the case of the virus trapped by mature dendritic cells, these viruses can be efficiently transferred to bystander CD4 T cells that become productively infected through a mechanism that is, first of all, highly infectious and is known with the term of transinfection because it requires the transfer of captured virus from the dendritic cell to the T cell. In our laboratory, when we use lipopolysaccharide or interferon alpha to mature dendritic cells in vitro, and you have to remember that these are all factors that are upregulated in the plasma of HIV-infected individuals, what we see is that these mature dendritic cells have an enhanced capacity to capture HIV. And indeed, this is just a live cell imaging movie in which we have used a green fluorescent tag virus to follow the trafficking pathway and capture by a mature dendritic cell. And what you can see here is that in less than a few hours, this mature dendritic cell is able to capture and store large amounts of fluorescent virus that is stored within a sac-like compartment. And just in a few hours, a lot of viruses are trapped within this cell. And importantly, these viruses can be efficiently transferred to target cells. And indeed, when we compare the ability of mature dendritic cells to transfer the infectivity to a T cell, T cells have a much more potent capacity than immature dendritic cells to perform this transinfection mechanism. And this has been reported by many different groups in the literature working with many different kinds of models for dendritic cells, but also primary cells. So overall, what we believe is that within the lymphoid tissue, mature dendritic cells could be playing a key role mediating the infection of new CD4 T cells through this specific mechanism known as transinfection. Initial studies related transinfection to the ability of several C-type lectin receptors. I think that the most famous one is Disisign, and these are all receptors that are expressed by dendritic cells on their surface. The idea was that transinfection was dependent on the ability of these receptors to specifically interact with the envelope glycoprotein of the virus. However, we in our lab were able to show many years ago that when we were working with viruses that lack the expression of the envelope glycoprotein and therefore could not be recognized by these C-type lectin receptors, these viruses were captured as efficiently as wild-type viruses, indicating, first of all, that alternative receptors, aside from C-type lectin receptors, should be implicated in this internalization mechanism, but also that alternative viral factors, aside from the envelope glycoprotein, should be playing a key role in this initial recognition. Which are those viral factors then? Well, we turn our attention into several lipid components that are integral part of the HIB membrane, and in particular, we look at gangliosides, which are lipids that are anchored to the HIB membrane but expose several sugars in the outer part of the membrane and are sialylated, therefore they have sialic acids. And in particular, when we remove gangliosides from the lipid membrane of HIV, what we observed was that mature dendritic cells were no longer able to recognize the viruses. And this is what you're seeing in this part of this live cell imaging movie. We have used viruses that lack gangliosides on the membrane, and therefore mature dendritic cells are not able to capture and store these viruses. There is no compartment being formed, as opposite to what happens when we use wild-type viruses that do have gangliosides on their membrane, can be recognized and can be internalized within mature dendritic cells. Using this type of approaches, we and others have been able to identify gangliosides as the key moieties that are anchored in the lipid membrane, allowing the recognition of mature dendritic cells and allowing the internalization of HIV within these cells. And knowing these key moieties is much more easy to address which receptors are implicated in this mechanism. We started by performing comparative transcriptomic analysis in which we compare highly transinfecting mature dendritic cells with other dendritic cells that have a lower capacity to transfer HIV. And it became very evident that the Siglec-1 expression was highly predominant in those cells that had a higher capacity to transinfect. And indeed, this Siglec-1 receptor belongs to a family of I-type-lectin receptors that can bind sialylated molecules such as the gangliosides that are anchored to the HIV membrane. So this gave us an idea that this could really be the receptor we were looking for. But in order to test this hypothesis, we performed a series of functional analyses. We started by using specific monoclonal antibodies against Siglec-1, and we added these antibodies to mature dendritic cells before adding exogenous virus to the system. And what we saw is that when we use these specific monoclonal antibodies against Siglec-1, we could nicely downregulate and diminish the capture of HIV by these dendritic cells, something that we did not observe when we use an isotype control or when we use specific inhibitors that would target the C-type lectin receptors, which is the blue case here. Importantly, when we follow the trafficking route of captured virus within mature dendritic cells, and this virus can be seen here in red, what we observed was that there was a beautiful colocalization with the Siglec-1 receptor, shown here in green, and therefore that's why you're seeing this yellow compartment within the mature dendritic cells. In addition, we were also able to confirm these results by rescuing the transinfection capacity of an antigen-presenting cell line that lacked the expression of the Siglec-1 receptor just by transfecting this cell line with a plasmid that specifically codes for Siglec-1. And as you can see, when we did so, we could nicely rescue the ability of this cell line to transinfect HIV through a mechanism that was specifically blocked by a monoclonal antibody against Siglec-1 and through a process that we never observed when we use plasmids coding for other members of the Siglec family. Moreover, we were also able to detect the expression of the Siglec-1 receptor within the lymphoid tissue of HIV-infected individuals. And indeed, Siglec-1 positive cells localized within perivascular and perifollicular regions of these lymphoid tissues, where CD4 T cells are also localized, indicating that in this anatomical compartment, transinfection could be taking place. But what happens if we are working with primary myeloid cells directly isolated from lymphoid tissues which are expressing Siglec-1? Until this point, we had only worked with dendritic cell derived from monocytes or derived from blood, but in these particular experiments, we wanted to work with primary cells directly isolated from tissues. And what you can see here is that these cells that express Siglec-1 can nicely capture fluorescent red viruses, and you can see the compartment being formed within these primary myeloid cells coming from lymphoid tissues. And moreover, these cells have the capacity to transfer infectivity to CD4 T cells that become infected through transinfection, and this mechanism can be specifically blocked if we use a monoclonal antibody against Siglec-1 to pre-incubate these cells before adding the virus. So overall, this data led us to believe that Siglec-1 could be a key receptor mediating transinfection within the lymphoid tissues of HIV-infected individuals through a mechanism that clearly begins by the recognition of gangliosides that are anchored to the HIV membrane and which are nicely recognized by Siglec-1 receptor located in myeloid and mature dendritic cells within these secondary lymphoid tissues. So I think it's quite nice and it's really beautiful because these in vitro data have been recently confirmed in vivo by an independent group working at Yale University, and they were able to confirm all these results in vivo by working with the murine leukemia virus as a model system of retroviral infection in mice. And the work of this team was based on our observations that gangliosides, viruses having gangliosides on the membrane, such as the MLB, would be nicely recognized by the Siglec-1 receptor. The work of this team was published last year in Science, and they had been the first ones showing transinfection taking place in vivo within the lymphoid tissue of an infected animal. And here, what you're observing, I'm going to play the video in just a few minutes, but what you're observing are Siglec-1 positive cells that have huge amounts of the murine leukemia virus, which is shown here in green. And what you're going to see when I play the video is how this virus is transferred to the target cells, which in the case of the MLB are B cells labeled in red, and this is taking place within the lymphoid tissue. And here is the transfer of the virus. And this has been imaged in a living animal. Also, data which are very significant coming from this group as well is that when they work with wild-type animals, they can detect the murine leukemia virus within the lymphoid tissue very nicely, but the key experiment here is that when they use Siglec-1 knockout mice, the viruses are drastically reduced within the lymphoid tissue of the infected animals. So I think that the important question here is how can we translate all these in vitro and animal studies into HIV pathogenesis in humans? What are the implications of these data I've just shown you? This is not a very easy question to answer, but we've tried to address it by looking for specific genetic variants that could lead to the truncation of the Siglec-1 receptor to the absence of expression in humans, and could allow us to test what would happen if these individuals lacking Siglec-1 receptor would be infected with HIV. So we started a collaboration with the group of Amalio Telenti, and we identify all the possible truncating variants that could affect the Siglec-1 gene, and in particular, we focus our attention on this very specific truncating variant. It's a stop truncating variant that has a very low frequency. It's only 1%, but still it was high enough to let us think that we would be able to find this variant if we look into large cohorts of HIV-infected individuals. So we screened more than 4,000 people, and we were able to identify 2 homozygous individuals carrying this particular variant that could possibly lack the expression of the Siglec-1 receptor, and 97 heterozygous individuals that could possibly express a reduced amount of Siglec-1. To confirm this genetic data, we were lucky enough to have cells coming from these different patients in which we were able to measure the amount of Siglec-1 expression. And what you can see here is that while wild-type individuals have a very nice expression of Siglec-1, this expression is reduced in heterozygous individuals, and the expression of the Siglec-1 receptor is totally absent in the homozygous individuals bearing this particular stop variant for the Siglec-1 gene. This clearly indicates that we had found Siglec-1 null individuals within this cohort of HIV-infected individuals. So, importantly, the lack of expression of Siglec-1 correlates with the lack of capacity of these cells to capture HIV and also to transfer the virus to target cells. And this is what you're observing in this data. Wild-type individuals express Siglec-1 and can nicely capture the virus through a mechanism that we can block with an antibody against Siglec-1, and that is not the case for the homozygous individuals lacking Siglec-1 expression, where there is no capture of the virus and therefore there's no transfer to the T cells. So we started then to look a little bit closer into the clinical parameters of these different groups of individuals, and we were not able to find significant differences in terms of viral load or in terms of CD4 dynamics over time. But when we look at the time of survival free of AIDS within these distinct populations, in the absence of antiretroviral treatment, we found a trend for those carrying the Siglec-1 stop variant, which were somehow developing AIDS or progressing less obviously than the rest of the individuals, the wild-type individuals expressing Siglec-1. We had seen this observation only if we focused in patients for whom we know the exact seroconversion date, but also if we look into the whole cohort. But I should mention that the difference or the trend that we observe in this graph is not statistically significant. And the problem here is that we would need an extremely large cohort. We would need over 10,000 individuals in order to get solid data and confirm these results that with a cohort of only 4,000 individuals, we cannot really confirm. So overall, I think that we clearly believe that Siglec-1 could be a key mediator of the infection of new target cells within the lymphoid tissue, but further studies are required in order to dissect the contribution of this receptor to this mechanism. And in that sense, I think that the identification of individuals that lack the expression of Siglec-1 in nature, because they are null Siglec-1 individuals, tell us that this receptor could be a nice therapeutic target that might not have important side effects. And we are trying right now to develop a family of novel compounds that could block the Siglec-1 receptor HIV transinfection through this mechanism. And I think that maybe these tools could complement some of the eradication strategies that right now are aimed at reducing the size of the viral reservoir. And I would like just to finish by acknowledging all the people involved in this work, the HIV cohort study, and the group of Amalia Talenti, also Mary Carrington, and all the people from the MAX cohort that allow us to confirm the results obtained in the Swiss cohort, the groups of Oliver Kepler and Hans-Jörg Klauslik at Germany that were key in identifying Siglec-1 receptor and gangliosides as the viral moiety. Also all the physicians in our hospital who provide us with tissues and pathology samples, and of course the many, many people within the group who have been working for the past decade in this area of research. Thank you very much for your attention. Thanks a lot.
Yes, estupendo. I have two questions. The first one is, in this process of capture and trapping of the virus by mature dendritic cells in the process of transfer, do you think that in the same compartment there is a sensing by TLR7 or 9, for example?
That's a difficult question to answer. We have data coming from transcriptome analysis in which we don't see much differences in terms of upregulation of genes after viral capture, maybe just 4 hours. Maybe we would need longer time in order to measure those changes, but it's nothing as striking as adding LPS, for instance. So it would be very subtle changes. Yeah.
Okay, and the other question is, well, it's just for me for my knowledge. You know, so what is the balance between Siglec and this design? I mean, do you think that in your hypothesis, for example, in mature dendritic cells, this design could be still have a role? I mean, your data on the null patients, in which there is no transinfection, points towards a major role for Siglec-1. Do you think there is a balance, or is really a predominance of one?
In our studies, we've worked with immature dendritic cells as well, and the contribution of Siglec-1 in those cells is the same as this design. And we do see an effect. We can block HIV transinfection by immature dendritic cells using inhibitors of this design by 20%. But the problem is that immature dendritic cells do not transinfect virus as efficiently. I mean, the fold change, the differences comparing both types of cells is, you know, mature dendritic cells have maybe 100 or even in some experiments, even 2 log differences in terms of the capacity of transferring the virus. So yes, this design might play a role in immature dendritic cells, but the extent of transinfection is so limited that I do not think it would be relevant in the context of lymphoid tissue, where most of the cells would have a mature phenotype. That's my guess.
That could be important for the transport of the trapped virus from.
I don't know. I mean, some of the beautiful studies performed by groups working with non-human primates have shown that PDCs are some of the first cells that arrive to those tissues, and they would be secreting interferon alpha, and maybe within those tissues there would be some activation there, allowing mature dendritic cells within those tissues in the migrating pathway, capturing viruses and then moving towards the tissue. So I don't know, it would be both pathways.
Luis?
I think, have you had the opportunity to look at the Siglec-1 in macrophage? We talked about this a little bit, but I was thinking, you know, the CD4 molecule is very low in expression, and it also recirculates as opposed to being locked in the cell membrane for the T cell. And to the extent that Siglec-1 could be a strategy by which the macrophage could actually acquire the virus and perhaps be infected more efficiently as well. I mean, it's a different type of question, but.
Yeah, there's several groups which have observed that macrophages express Siglec-1, and that the interaction with this receptor is leading to an enhanced infection of the cells. We haven't really done that because our work has always been focused on the transinfection pathway. But in terms of transinfection, when we work with macrophages derived from monocytes, what we observe is that in contrast to what happens with dendritic cells or monocytes, the virus that is retained by these macrophages via Siglec-1 is very efficiently degraded. So maybe it could be a mechanism that could lead to antigen presentation rather than contribute to the infection of new target cells.
You don't see any enhanced infection?
No, there is transinfection, but then again, it's extremely limited, because in this case, Siglec-1 is leading to a very rapid degradation of the capture virus. So it's a different setting, and then again, we would love to work with primary macrophages coming from tissues in order to confirm these in vitro observations working with monocyte-derived macrophages.
Any other question? Okay, thank you very much for these 3 fantastic talks, and to all of you, thank you very much.
Yo quería venir de Izquierdo a charlar. Gracias. Es que si empieza a haber charlas de estos niveles, al final es eso, hay que hacerlo en inglés. Porque